It's a pleasure to introduce Brian Berkowitz today. Brian received his PhD or BSc in technology and has been in Weizmann since 1993. He received multiple awards, most notable he's an AGU and GSA fellow. He'll talk today about transport and fate of nanoparticles and pharmaceutical compounds in soil water environments. Just to let you know, next week's seminar will be given by Yair Cohen from Caltech, and he'll be talking about tropical cyclones. Thank you, Brian.
Totally different subject. Anyway, thanks very much. It's been a long time since I've been here. I mean, a really long time. So it was nice to get the invitation to come back. So the talk today, I was just saying to Simon, it's kind of science light, because the first half of the talk or so is actually a historical review of emerging contaminants. I got into this and said, hey, you know what, we really don't know what we're doing, and so before we do the science, let's see what the background is. So I'll be talking about pharmaceuticals and nanoparticles and some more general things as well. And my main colleague over the years is Ishai Dor. He's a staff scientist in my group. And I couldn't even name the number of students, because all of this is work or things that have sort of affected our thinking, my thinking over the last probably 15 years or so. So many, many students, I won't mention names, but know that this is not a sole effort. By the way, if there are questions or as we go along, please stop and ask me, because it's more fun that way too. So before we talk about specifically pharmaceuticals and nanoparticles, they belong to the general class of what are called emerging contaminants, okay, or contaminants of emerging concern. And the formal definition is any synthetic or naturally occurring chemical that's not commonly monitored in the environment, but has the potential to enter the environment and cause various adverse effects, either ecotoxicological or toxicological to humans. And you see through history, we invent something new, we say, wow, this is great, this will save the world, this is everything we need, it's released accidentally, intentionally, whatever, we're exposed to it, and afterwards we say, Gewalt. What have we done to ourselves? Look at the harmful effects. And as I say, in particular, we're looking now at nanomaterials and pharmaceuticals, but it's fun first to go through emerging contaminants. They're now not emerging, but if you look past, present, and future. So we're looking to the future, fluorinated materials, hybrid cars, what are called technology-critical elements. In every hybrid car, there are between 10 and 15 kilograms of rare earth metals. Okay, we know almost nothing about them. And for the geochemists in the audience, if you talk about neodymium, indium, gallium, thallium, ytterbium, europium, all the metals at the bottom of the table, you know almost nothing about them. Okay, there are no thermodynamic data, how they behave in the soil water environment, when they're released, and they are being released. We're just now beginning to investigate how these materials behave. Thallium, we know, is toxic to humans. It's used in all kinds of materials. Every cell phone has got, several micrograms of indium and gallium. Touch screens are all with indium. All these materials, as I say, are being released. We've got no idea what the effects are. This is to the future. We're looking currently at pharmaceuticals and nanotechnology. Let's go back in history. What destroyed the Roman Empire, among other things? People say lead in the water, right? It led to nervous problems, dementia. The concentration of lead in water was expected to be, or it was thought to be as much as 100 times what was locally available. But it turns out another major source of lead was in fact the first artificial sweetener. We talk about sucrosite and stevia and all these other things. It turns out the first artificial sweetener was lead acetate, or sugar of lead. And the Romans intentionally, they discovered that lead is sweet. They took pots with lead, filled them with wine, and boiled them down to get a really sweet liquid. And they would add this to everything. And there were estimates they would drink as much as 1.5 liters of this sweet liquid every day. So then they realized at some point, lead is probably not so healthy, but they were addicted to it. It was convenient, it tasted good, so you know, you have to say that. We don't need to worry, it'll be okay. So this was actually a major source of lead poisoning in Roman times. Go forward a little bit, or just before that, do you know the expression in English, mad as a hatter? You know, from Alice in Wonderland, remember the Mad Hatter? Do you know where that comes from?
Mercury.
Sorry? Mercury from making the hats in England. There was mercury chloride, I think it was. They were inhaling the mercury, using the mercury to form the felt and the cotton to give the hats a shape. They all suffered from cancer. They all went crazy. So the next one then is you look at radioactivity. Marie Curie discovered radioactivity in 80? 18 when? 1904. I thought already there was syndication before that. Okay, 1904? Okay. So by 1920s and 1930s, there was a French company that became then a British company that was then a French company that was called Floradia. And said, gee, you know what, radium is great. So they were radium pendants for rheumatism, radon water, and radium in water was the fountain of youth. You had uranium blankets for arthritis, and thorium was terrific for digestion. And you can actually go through, I found a bunch of advertisements. This is great to read. Here's health. I'll just read this. It says, to keep our health, to keep that health, you must keep nature's law. They're simple and easy to keep, but if they're broken, nature exacts a heavy penalty from each and every one. Get plenty of sleep, exercise, and wholesome fun. Avoid overwork, all other excesses. Eat fresh, natural foods. Breathe fresh air, and drink plenty of fresh, invigorating, natural radioactive water from the radium spawn. This is a real ad. There's another one. Radiosoil. Certified radioactive water. This was the fountain of youth. Okay? Now we laugh at this now and you say, how could people have been so incredibly stupid? The concern is, are we doing the same thing to ourselves now with other wonder drugs and chemicals and all? Okay, and this isn't to start panicking everybody. I'm raising the question. Okay, not more than that. This is great. It was radium toothpaste. And in Germany, 1930s already, you had chocolate bars laced with radium. Eat a chocolate bar a day, you're supposed to look younger from that. Okay, this is serious. Floradia was the... Sorry?
Certainly dia.
Yes, yeah. And in the 1930s, the Nazis already knew about radioactivity for other purposes. So they knew on one hand it kills you, on the other hand, it's the fountain of youth. And you look at these cleansing milks, powders, and everything else. The first use that I found of radium was in lip gloss. It was known that radium kills bacteria. So if you put it in lip gloss and put it in your bacteria, that's good, right? It kills all the bacteria. Of course, it kills everything else, too. So we just put this in here. She's glowing, but it's not because of beauty. These are serious materials. They were used. Then let's move forward a little bit. DDT. Remember the wonder chemical DDT? It was first discovered in 1874. It was used after World War II. Cases of malaria dropped from over almost 500,000 at the end of the war to none by 1950. Okay, malaria was wonderful in terms of killing off the mosquitoes and carriers. If you see these were real advertisements. Look how good DDT is. Spray yourself with it. And this is terrific. DDT is good for me. These are advertisements where the ladies know what's good. So you're supposed to spray in your child's bedroom. Spray it with DDT. It'll get rid of all the pests and everything else and keep your child healthy. And the advertisements here, it's good for fruits and vegetables. They even at one point they said spray it on your food just to make sure that you've got all the insects are killed. We banned it, the world banned it in 2001 because it's persistent. It's thought to be carcinogenic. It turns out now, relative to atrazine and many other chemicals we're using, DDT is fine. Okay, these things are all relative. But once something is banned, try to bring it back again afterwards. Okay, so we all know DDT is bad. So you look at these ads. So now moving ahead, now what are the new things? We've got nanomaterials and we've got pharmaceuticals, and so I'll talk about these two different directions. So first of all, what are nanomaterials? The formal definition is that at least on one size of the particle, it's less than 100 nanometers. Okay, that's the formal definition. People who used to think of the micron scale, talcum powder, things like that, that's the micron scale. The difference between a micron-sized particle and nanoparticle, do the calculation, it's the same as between an elephant and a ladybug. Okay, so that's the neat thing. They're really, really small particles. First thing to point out then is you have surface reactivity. So if you think about a sphere the surface to volume ratio, as you decrease the shape, you have a much higher surface to volume ratio. So 1 elephant versus 1 trillion ladybugs, the surface reactivity, the surface area here is of course much, much larger. So per gram or per kilogram, nanoparticles are going to be much more reactive. So these are old statistics. This is from 2012. It's a huge market. It's growing even more than exponentially. We're finding nanoparticles in just about everything now. Now, one of the major uses is in health products. All of the face creams and cosmetics that men and women use, they go on much smoother than they used to, okay, because they're full of nanoparticles. Sunscreens is what the first actual use of nanoparticles was sunscreen, titanium dioxide. Now, it's marked here that they're not always truthful. If you look at a container of sunscreen, maybe it'll say contains titanium dioxide. That's all it'll say. It won't say nanoparticles, nothing more. Legally, they're covered because it says may help reduce the effects of. Okay, it doesn't say prevent sunburn or anything else. May help reduce the effects of. Now, what's titanium dioxide? For the chemists in the audience, it's a photocatalyst. Right? It breaks down DNA, it breaks down bacteria or organic matter. Some people do here geochemistry, catalysis, right? Okay, so we know titanium dioxide, photocatalyst, breaks down material. So when you first started with the sunscreen, you put on your skin raw titanium dioxide. So it reflects the UV light. The question is, what does it do to the skin? So after a couple of years, and the FDA approved this, afterwards they said, well, this isn't so healthy, so they started coating the sunscreen. You know what the first coating was? Benzene. Okay, so now, how is this allowed? Okay, so the FDA allowed that too. A few years later, the companies realized that wasn't so healthy, so now they use some silica coating. It's not clear, it's proprietary. Presumably it's safe, but that was one of the histories. So you have nanoparticles in all kinds of products. In the health industry, silver in particular is an antimicrobial, antibacterial agent, anti-odor, so it's used in, where was it? I have a picture somewhere showing it. In sports clothing, socks, all the sports clothes now are all laced with nanoparticles, which means every time you wash them, they're released. They're used in paints. Zinc oxide is shiny. Okay, it has what do you call little sparkles to it. So it's used as a whitener. I'll come back to this in a moment. So in a whole range of products, we're finding nanoparticles. But again, usually it's not indicated what they are. So we have quantum effects, we have huge surface areas, and the question is, what is the effect on the environment, on ecosystems, and on human health? We know we've got increased reactivity, we know there's enhanced toxicity, we know there's high mobility, certainly in the human body there is. We're looking in other organisms in the soil water environment. I'll discuss that. And in terms of the pathways, manufacturing, transport, use, they're released all the time. Improper disposal, intentional, accidental release by companies. We see this all the time. And again, in cosmetics and personal care products, every time you shower, whether it's once a week before Shabbat or once a year in Yom Kippur, whenever you shower, all of those nanoparticles are released, into the wastewater. And we know in wastewater streams, if you look at the Shaftan project in Israel, most of those nanoparticles remain. Okay, they stay in the water, and again, they're released onto the soils. There's a question about the mobility. In principle, they're so much smaller than large particles, we expect them to be very, very mobile. And we'll talk about that in some of the experiments we're doing. They're huge, because of the large surface area, we know for sure you can have other contaminants that then sorb or are retained on the nanoparticles and are transported much faster. Okay, so if you have a normal organic material that would absorb to the soil, if it absorbs to the nanoparticle and it moves really quickly, then you move that material further downstream than you would have predicted. Reactivity, we're still trying to figure out what's going on, and we get then bioaccumulation. So there are all kinds of results showing you get stunted plant growth, silver kills all kinds of things too, bad and good. And I'll show some other examples afterwards, some of the other effects. Health risks, toxicity, we're not sure about. I mentioned before about titanium dioxide. That's on the nanoparticles on skin, for example. You get free radicals, DNA damage, other breakdown. There's some strange things going on. Carbon fullerenes, that's the buckyballs. C60, the buckyballs, they're used now as lubricants. They don't break down. They have all kinds of effects on fish in particular and smaller aquatic species. I don't know if it's changed, but until 2 years ago, the U.S. Environmental Protection Agency had the strange rule that if you're working in a laboratory and you have leftover organic material, of course you can't dump it down the sink, right? You have to go to organic waste. Buckyballs, the C60, well, that's just carbon. Carbon's perfectly safe. You can dump it down the sink. So even from the regulatory point of view, there's not the awareness that, sure, it's just carbon, but because it's this particular structure that doesn't break down, and we know it has toxic effects, regulations have to change. They're taken up by membranes, and once in the bloodstream, we know they move very well, and nanoparticles gather on the interfaces of organs in the human body. Okay? So the toxicologists are having a heyday with that right now. So where do we have a lot of question or some strange things from a regulatory point of view? We had a patent some years ago where we were using nanoparticles to treat contaminated water. We were able to break down a whole range of halogenated materials. We did field tests with the Israel Water Authority, tested with the Ministry of Health. We had heavily polluted water from wells that were closed. We turned it into drinking water. We bought a company, we want to market. The US EPA says, no, you can't use any technology that has nanoparticles in it because we're afraid of them. That was 10 years ago. The laws are now changing, but you still have a very difficult regulatory process to use nanotechnology, anything related to drinking water. On the other hand, you can inject nano zero-valent iron here underground. You can have a pumping well 1 meter away, and that's fine because you're pumping groundwater. Okay, the fact that they're going to be nanoparticles and that has no effect. So that's the EPA says you're not allowed to drink nanoparticles. The FDA, the Food and Drug Administration, says, oh, you can eat them, there's no problem. So right now, marshmallows, icing sugar, zinc oxide, and copper oxide are sparkly. So it's in the food products and you're allowed to eat them. Okay, so this is still a law right now. You can eat nanoparticles, God forbid, don't drink them. Okay, and this is, as I say, when you talk to the regulatory agencies, it's very frustrating, they don't get it. Okay, the zinc oxide, by the way, also it's a whitener in toothpaste. I think I've got toothpaste here somewhere. All the whitening for teeth, that's all zinc oxide. So this is what we're not sure about with nanoparticles. Some of the effects of nanoparticles, this is some other experiments we done a few years ago. If you just take corn, that's a normal corn seed just after germination. If you expose it to bulk copper, forms of copper, and ionic copper, you get some changes, but those are still healthy. The moment you expose them to the nanoparticle scale, and you start germinating with nanoparticles, you get all kinds of deformations, and it messes them up. These are some other pictures just showing a healthy leaf and the exposure to copper oxide nanoparticles. There are serious effects there. That was just one example. Some experiments we did recently, about 2 years ago actually. We took 2 kinds of soil from the interior forest in the south, baked gone soil. Those are just EM images of the controls, exposure to 1 and 5% of the copper oxide nanoparticles. You can see you get aggregates, and then by the time you get to 5%, you get coating on the soil. So all of the reactivity or the absorptive capabilities or cation exchange to do with the soil here is altered drastically when you have nanoparticles, plus you get additional reactivity. So we worked with Dora Mintz at the Volcani Center, and you can do what's called DGE fingerprinting. You're looking at the RNA fragments, and you've probably all seen these I just call them barcodes. Okay, nobody really understands these things, but the only thing you need to understand from here is this is the baked gone in the interior soil, the control after 24 and 48 hours without any exposure to nanoparticles. After exposure to 1% magnetite and copper oxide, you can see this type of bacterium has been destroyed. It no longer exists. We now facilitated the growth of other ones. Okay, so wherever you see changes here in these bars, it means there is a change in the microbial community. So we know exposure of soil to nanoparticles changes the microbiome, what's in the soil there. The very upsetting thing to me is that we don't have a clue if this is good or if this is bad or what the effect is. In a cubic centimeter of soil, you have something like 10,000 different species of bacteria. Okay? Of those 10,000 species, if you can culture 6 or 7 of them, you're lucky. Okay, the rest you can't identify, you can't culture. All you can do is look at DNA strands. So we know, again, we know there's a big change in the community here, but what we've actually done to it, we don't have a clue. Okay, so we answered one question, we are now left with many others. So this just leads me, I found this cartoon a long time, so very often we really don't know what we're supposed to be looking for either. This cartoon kind of sums it up. When we go to the lab and say, well, nanoparticles, pharmaceuticals probably have some effect. What should we be studying? So that's nanoparticles. We'll come back to that. Pharmaceuticals. Now you've heard about a lot of this in the literature and in the news. Has Benny Heffitz lectured here recently? He's doing a lot of work from the Faculta.
Last year.
Last year, okay. So he's also doing work on uptake of pharmaceuticals in carrots and red peppers and whatever else. So the simplest way to explain what's going on is the human body is not very efficient in terms of dealing with various medicines. You have to have a high concentration in the bloodstream to cross the cell barrier, but as much as 90% of all the drugs we take exits the body in the urine with essentially no change. Okay, so take essentially 90%, 80-90% of all the drugs, flush them down the toilet. From there, they go to the shutdown, to the water treatment facilities, which generally do not remove these materials. Now, work has been done to try to figure out what's the life cycle of the pharmaceuticals. We're here in the center, and you look at a figure like this and say, what a mess, and it is a mess, and nobody understands it, but you find all the sources of where it goes and whatever else. The simplest way to look at it is that. Okay, so these are the efforts that we made. It's not just human consumption. The other major consumption is poultry and cattle growth for dairy farming. Growth hormones, antibiotics, they all come out. In the case of chickens, it comes out, of course, in the manure, which is used as a wonderful fertilizer. And once it's, of course, in the fertilizer, again, 90% of the hormones, antibiotics, are now in the fertilizer, which finds its way into the soil. Okay? We're doing this to ourselves. By the way, there's another one we were working with. I don't know how the farmers discovered this. There's an arsenic-based material called roxazone that's given to chickens. It increases their appetite. Okay, I don't know who discovered give chickens arsenic and they eat faster. Okay, they eat more. So we now have arsenic also being in the soil or being released into the soil. We're able to trace its movement. Okay. So we have the sources from here as well. So now when we start testing water, this is tap water in a whole range of countries. You name the chemical or the compound, and you find them in various concentrations. Now, a major question that nobody has an answer to right now, we all know, or we're supposed to know, that drinking water standards for organics and heavy metals in water is maximum 4 parts per billion, unless that's been changed. Right? That's the European standard, the North American standard. It's supposed to be the Israeli standard. Chaim, you know better than me. Is it followed? Odlo. Okay. So the maximum is supposed to be 4 parts per billion. Okay. But we don't know with pharmaceuticals if 4 parts per billion is a concern. Less than that is a problem. More than that, we don't know yet. We're exposed to it. There's all kinds of circumstantial evidence. So if you look around the world, we have declining fish populations. Okay? There are 2 reasons for declining fish populations. One is overfishing. The other, it turns out, in industrialized countries, there's feminization of the fish. There are not enough male fish in lakes and rivers. Okay, why? It's not known. But when you test the water and you find 100 parts per trillion or 1 part per billion of estrogen at low levels, so it's bioaccumulating, it's there all the time, and you have feminization of fish, that seems to be an indicator that something's going on here. Okay? In humans, too, there are cases, certainly in North America, of menstruation of, they're not even women, of girls, begins as early as the age of 8. Okay, it's happening at younger and younger ages. And we don't know why. Now, again, there's something like between 60 to 100,000 anthropogenic materials that are being produced every year. So if you get cancer, is it from something in the air, something in the food, something in the water, combination of materials? There's so many confounding variables, you can't identify what the particular cause is. So this is another problem that there's a lot of circumstantial evidence. But again, anywhere you test, So this is part per nanogram per liter, so that's part per trillion level. In some cases, you've got very high concentrations. It also gets fun because you can look at recreational drugs. Some colleagues of ours in Spain were looking at the Barcelona water supply. So again, you can measure the part per trillion level in wastewater. So you look for cocaine, for example, and you can find what's the concentration in the wastewater. Then back calculate from the volume of wastewater how much cocaine entered the water supply. And during the weekday in Barcelona, there are 14 hits per 1,000 people of cocaine. And it spikes, in fact, on the weekend. Okay? There are no secrets there. We know this. Ecstasy, 4 hits per 1,000. That's not just Barcelona. This was, where was that? The Berkowitz in, where was that? In Germany. Okay? So you have, again, detectable concentration throughout. And I was in Italy last year, and I knew I had this information, I presented this, and I knew they'd just say, oh, that's all those damn Spaniards, they're on drugs all the time. So I checked the statistics in Milan, 4 kilograms of cocaine enter the Po River every day. Okay, so that's the fun part, too, no secrets. So when I give a popular talk, I always like to take this picture from Hitchhiker's Guide to the Galaxy, because by now people are saying, my God, what do? I can't drink the water, I can't eat food, and I can't breathe the air to stay healthy. So the other thing to point out is that, again, it's all true except for the bits that are lies. Okay? What are the lies? I know we're still trying to figure it out. Okay? So we know we're being exposed constantly to presence of hormones, of chemicals, of nanoparticles. What are the toxicological effects? Don't know yet. So we're at the stage right now where we have to understand, we have to monitor, understand what the processes are, and then try to shed light on that for future analysis. Just there was one paper or one presentation I saw last year that was fascinating, just to give an idea how very small concentrations are really critical. There was a study of rat embryos. Okay, so every litter of rats has 10 to 12 embryos that are growing or that develop. And so what they did was they would measure a female that developed between 2 other female embryos, and a female that developed between 2 males. Okay, very simple. They did this over many, many repetitions of studying different rats. And invariably, the female that developed between 2 females when it was born was much more passive than a female developing between 2 males. Okay, so these are not even picogram liter concentrations, differences in hormones. Really, really subtle differences in hormones, just what they're exposed to in utero, and that already affects. So is 1 part per billion of estrogen or something else in effect? Probably. But we have to prove it. So the scientific part of what I want to talk then, or the more scientific part then, is looking at some of the experiments we've been doing to understand how nanoparticles and pharmaceuticals behave in the soil water environment. So the first problem is detection. If you take, 100 milliliters of double distilled water and add a known quantity of estrogen, and then check it afterwards with HPLC or GC or LC, whatever, you know what you put in. It was super clean water, very easy. Now take a soil column, run water through it with various chemicals, and you get all this schmutz coming out. There's no, by the way, in Germany that's a technical term. Schmutz is the allowed term. So you get, sorry?
No, it's schmutzig.
Schmutz, yeah. So you get the schmutz coming out. So try to identify from that, do you have nanoparticles? How do you separate the nanoparticles from the dust and the other colloids and whatever else is in there? Try to detect the pharmaceuticals in that. This is really a major problem at all levels, especially when you're trying to get down to the PPT level. Afterwards, we have to consider all the transport, modeling, geochemical reactions. There's one surprise I'll explain afterwards if I have time. So there are a lot of things to be looking at. So first, in terms of detection, this is work we did with Giboa Arya a few years ago. This is, again, looking at the Shutdown project. And in the good old days, we were able to use tritium. Tritium was a wonderful tracer to look at groundwater age, right? We were what would they call it? Not nuclear bombs, nuclear devices. That was the atmospheric testing, and most of it was going on again in 1963, 64, I think, didn't it? I think that was, yeah, give or take. So you have all this radiation, you have tritium being formed. So as a byproduct of that, tritium was a great tracer for groundwater. We know when it came down in the rainfall. Actually, you did work on that years ago. That's right. So you want to give the lecture? So anyway, it was a great tracer. The half-life is what, 12 years? 13 years? 12 years. 12, yeah, okay. So by now, 50 years later, it's not such a great tracer for us anymore. So we're looking for other conservative tracers. So one of the things we find from the stuff down, you're probably all familiar with this, the soil aquifer treatment, the infiltration ponds and all. So Gilboa went and got information we tested the water there, and was coming up with these monitoring ponds, monitoring wells thereafter. And we were looking at carbamazepine. Carbamazepine is a muscle relaxant and anti-epileptic drug that's used very, very commonly. You can detect it at very low levels. And it turns out it's conservative. So carbamazepine now is a great replacement for tritium. We can follow it and see exactly where it goes. We've got all the wells there. We know what's coming in. We can measure the concentrations coming out of these wells. We've got a new conservative tracer. And experiments we did in the soil taken from different levels, you can monitor it quite nicely. It fits pretty well. This is bromide. The black is always the bromide. The chloride used as a conservative tracer. The red is how the carbamazepine behaves. Where there's more organic matter in the first few centimeters of the soil, there's a little bit of a delay here and some sorption and release. By the time you get down to half a meter already, it's conservative. It doesn't behave. It's not affected. It's a nice conservative tracer. So we talked about this with the Water Commission. Carbamazepine, as I say, is one of the new tracers we can use to find groundwater pathways. That's a convenient one. Then you get to something like estrogen. Now, there was a study done a few years ago in Germany where they said, okay, we took soil columns, injected estrogen, waited on GC to find the estrogen coming out the other side. 22 minutes, if I remember correctly, later, nothing appeared. So the conclusion was, we don't have to worry about the estrogen. It disappears. They didn't say what happened to it, if there was a metabolite, if it was absorbed. It just said, it's gone. So we reran the experiments, and sure enough, 22 minutes later, the estrogen didn't come out. But I told the postdoc working with me, I said, leave the LC running. Just go for a coffee, whatever. Let's see what happens. Roughly half an hour later, this huge D comes out. What is it? It took a few months to actually identify what it was. It turns out it's a metabolite of estrone, but it's estrone with a sulfate group on it. Okay. Where does it come from? Through the soil, whether it's microbiological or abiotic, I don't know. But we know now that estrogen then forms a conjugate with the sulfate. It turns out it's as physiologically active in the body as without the conjugate. Okay. So do we have to worry? Yes. In other words, at least we now know there's a metabolite formed, but it still moves through the soil, and this is very mobile. Okay. So that was one of these cases of detection that we came up with. So that was to do with pharmaceuticals. Now I'll spend some time on nanoparticles, and at the end I'll just say a few more words about pharmaceuticals. Now, nanoparticles are really annoying to work with, because you say, okay, they're small, should be easy. First thing we did some years ago, we took columns of glass beads. It doesn't get simpler than that. Glass beads, saturated with water, put the nanoparticles in. I thought this will be a no-brainer. We'll get really fast breakthrough curves coming out. We model them, we're done. After 35 pore volumes, that's as far as they've gone. They don't move. Okay, now on one hand you say, okay, that's the good news, right? They don't move. So if we now have a spill of nanoparticles, we don't have to worry. They're absorbed or they're stuck in the soil. There's no risk for contamination. Don't have to worry about it. Again, this is going back 2010, 2011. So then you say, just a moment, add humic acid. We know there's a lot of humic acid in the soil. What effect does that have on the glass beads? With humic acid, within a couple of pore volumes, it's moved through already. So, in other words, it becomes very, very mobile. And, and after 6 or so pore volumes, essentially the column was cleaned out. So humic acid and glass beads moves very quickly. That was with iron oxide particles. So, okay, let's see what happens with copper. Took copper oxide particles without humic acid, they move just as fast. Okay, so in other words, this is what the magnetite did, or the iron oxide particles with no humic acid. This is what the iron oxide did with no humic acid. The copper with no humic acid moves like this. Okay, so you can't even generalize and say all metal oxide nanoparticles are not mobile. Iron and copper behave very, very differently.
Did you change the pH?
No.
Did you check the effect on surface charge?
No, at the time we didn't do any of the chemistry done to look at surface charges, no.
I think it has to do with surface charge.
Yeah, exactly. Okay, but this is just pointing out, again, if you go back to 2010, the idea was all nanoparticles are the same, you don't have to worry about it. Again, that was what was in the literature. So,
in the literature, people are talking about the movement of particles. Well, they didn't term them nanoparticles at that time because the term was not available.
Yeah.
Microparticles or small particles. Everything is dependent on the surface charge.
right, but if you look at the literature on colloids, on larger particles, they behave differently. All the analysis that's being done, people are using colloid filtration theory, for example, to try to analyze how nanoparticles move, and it doesn't work. In fact, DLVO theory now has been shown it's not relevant, generally speaking, to nanoparticles. Okay, so I mean, that's a whole other issue. In other words, when can you use colloid or larger sized theory, what we know about them, when is it applied to nanoparticles and when not? And in many cases, it's a different ballgame. So, like on one hand, it should be obvious, but okay, this was in glass beads. This was a simple case. Oh, one other thing I want to show that. So then we thought also, so let's play further, fully saturated, and again, this is glass beads. What does the breakthrough look like once they start coming out? And one expects usually, if you look at the mass for a pulse of particles being put in over a number of pore volumes, you expect that you'll get, that kind of a nice pulse breakthrough curve. Add more humic acid, it becomes more mobile, so we get those kinds of pulses. There are many cases, though, with different salt concentrations, different pHs, this is different salt concentrations, same pH, where you get a pulse of material coming out, and then nothing, then another pulse, and afterwards another pulse. Say, how the hell am I supposed to model that? In fact, what's actually going on? The colloids don't do that as much. Okay? So what's happening with the nanoparticles is as they're moving through the system, they're aggregating, blocking the pores, so you get a change in the flow rate, and they block, and then the pressure builds up, and then they break through. Okay, so I can wave my hands and say that must be what's happening. We did a whole bunch of different experiments. Here, this is effective pH, by the way. Okay, so that's the salinity, that's the effective pH, and you get these pulses coming through. Do the experiment several times, and each time you get pulses appearing, but of course they're not consistent in terms of the when and the quantity because of the randomness. So you can understand the phenomenon, but to try and model that, it's not for me anyway.
It's good to understand.
Sorry?
Understand.
Yeah, so we can understand it, but to try to predict what exactly is going to be, we just say this is the phenomenon. So that was in glass beads. 2012, we then did some work. We said, you know what? We're all doing glass beads. What happens in natural soil? And we were at an AGU meeting in 2012. There were 20 presentations. 19 of them were glass beads, and ours was the only one with soil. And guess what we find? In soil, it behaves totally different than in glass beads. Okay. So one of the experiments we did, we took soil from, also from Betagon. It was filtered, or what do you call it? Sieve, into different sizes. So it's enough to know we had different pore size, or different grain sizes, or different fractions here from 1,000 microns down to 250, so we took 3 different pore fractions, took columns with each of these different fractions and said, okay, let's just take silver. Silver is supposed to be non-reactive, at least at the time thought to be non-reactive. And in the coarse, medium, and fine sands, here's the breakthrough that we get for a pulse. Okay, so you can see, and as you would expect, for the coarse, more material goes through. As you go to finer and finer sand, you get more trapping of the nanoparticles. Okay, that makes perfect sense. That wasn't a surprise. Then the question is that mechanical straining? Is it retention of some kind? What are the surface effects again? And trying to deal with natural soil with nanoparticles, it's a nightmare. I mean, try to figure out the chemistry on that. So that was the first effect. Then we said, okay, let's go back, test it on quartz sand, say with pure quartz. The blue and red and green, this is all bromide, which is 3 replicates of the conservative tracer. Here you have the silver as the crosses, and you can see here we have full recovery, 100% recovery there. So in other words, the retention here, I mean, this is some chemical, physical effect for the retention. But then there's something else we noticed. You expect, with the conservative chemical, after one pore volume, the average concentration should be a half. Right? That's where the center of mass moves through. Then we look back at this one, and you find that you have first breakthrough much earlier than one pore volume. So a portion of the silver is actually moving much faster than a conservative chemical would behave. Why? If it's repulsive force is negative charge, well, silver is positive. I don't know. But you have now an early breakthrough and these physical chemical effects going on. And then we say, okay, now let's look at the effect of the flow rate. As you might expect, higher flow rate, you get more material breaking through. Why? Because there's less time for sedimentation. That makes sense, right? A slow flow rate, the blue there is a much slower flow rate, so some of the nanoparticles can settle out, they're moving faster, but still you only get 40-45% recovery. So we've got humic acid, we've got the surface effects, we've got the flow rate. You can play also with the effect, again, this is showing the effect of humic acid in the column. Even with the humic acid, it's more mobile, and you still don't get 100% recovery, 80% as opposed to 40-50% without the humic acid. Lower the flow rate, and you get even less coming out. So this is, again, just to try to understand the basic phenomena. Again, how exactly humic acid interacts, that's still an open question then. And then silver, which originally was thought to be, well, we don't have to worry about it in the soil. We realized it actually forms, there are bridges, calcium bridges, and you get silver chloride forming. So we also took the soil and pre-treated it with potassium nitrate. So first flush it through with potassium. So this is the untreated. This is the recovery of the silver that we get. The moment you start flushing with more and more potassium, you now get higher recovery. You fill the sites, the reaction sites with the potassium, so the silver moves through, it doesn't react. So these are all aspects of the game. One more thing just to show, we just did a simple experiment. Take this is a suspension of the silver nanoparticles, add increasing volume or concentrations of salt, and here you can see there's actually a little aggregate of the silver that came out of solution, and here, it's very hard to see, here's actually the full aggregate. Essentially, everything comes out of solution because of the salt. Okay, so the presence of chloride has a positive effect in that respect. I should be finishing up soon, right? Okay, so I'm going to skip over that. Enough to say we did it in unsaturated soils as well, and we can model it. We've got a pretty good idea of general modeling. If we say, well, in this case, I'll just show one thing. We can say, if we say, just take a two-site model, so we have time-dependent and depth-dependent retention and release, we can actually model results very nice. Consistently, we're able to do that. But what exactly the retention processes are still remains open from the chemical point of view. Pharmaceuticals, everything I showed you for the nanoparticles, we find essentially the same kinds of behaviors for pharmaceuticals. There's also an added component, which they're a function of redox conditions. Okay, different redoxes behave totally differently depending on conditions. So in groundwater aquifers, that becomes a factor as well. And just one other thing I want to show, I mentioned at the beginning we have some geochemical surprises. We developed in the lab about these polymer beads where you add water to them, they have the same refraction index. Okay, so you have essentially transparent porous medium that you can work with. So we did a number of experiments here in the flow cell. We're injecting, this is what it looks like. We inject, this in this case was combo red, but you can inject nanoparticles, use a calibration curve, change the color intensity concentration, you get a map of the concentration from there. We did a whole series of experiments that I won't go into in the time. But what I wanted to mention here was we looked at positively charged silver nanoparticles. Okay? Now, the beads that we were working with, the porous medium is negatively charged. These had a citrate coating, they're positively charged. So we said, great, this should be a very simple experiment. You can see the beads here, exposed to the nanoparticles, they're attached, positive, negative, they don't move, that's it. So we thought we'll do a very simple experiment. You inject the nanoparticles here. By the way, that's interesting. Silver nanoparticles in water with the refraction come out gold colored. Gold nanoparticles, you know what color they are? Red. They're red, which is, I don't know, the first time I saw it, I thought, that's strange. Anyway, so these are the silver nanoparticles. you can see here the vast majority of them are captured, as you would expect. But we noticed after half an hour, there's actually a plume here. Some of the material is actually becoming mobile. What's going on? Now, when we looked at, we found, if you look at I'll just put it all in here. If you look at the zeta potential and the diameter of the particles, we actually, the inlet, if they were 182, the outer diameter, by the outlet, they're 3-4 times the size. So we're getting the aggregation of nanoparticles on one hand, but the other key point is if they started off stable and positively charged, within a short time, they're very stable but negatively charged. So we actually have charge inversion of the nanoparticles, which we really didn't expect. We did, again, at this point, didn't go into the chemistry behind it, but what it showed us also, and we've done other analyses afterwards, you buy from Sigma Aldrich, or you synthesize in the lab your bottle of nanoparticles, you think you know what you get. The moment you take the top off, you start to oxidize, put them in water, put them in soil, they change. Okay, so whatever you start off with, they change. In this case, sometimes within seconds, they change. So you have to look at those carbons. This is just to show we can model it very well so that we can understand. And so I'll end with conclusions where I don't actually have any conclusions. Okay, it's basically everything I said before. We're still trying to detect, we're still trying to monitor, we're still trying to understand what is the behavior of these materials. The key point is you have to do them in natural systems. In natural soils, under likely redox conditions, under real solution chemistries, because they behave very differently from sort of sterile, convenient conditions to measure. Okay, so I'll leave it open at that, and any questions. Thanks very much.
On the one hand, it's a very scary picture you go with here.
Yeah.
We don't know so much, there are so many parts. On the other hand, the life expectancy is increasing at a phenomenal rate of about 8 years per 30 years.
Yeah.
So maybe there's no room for concern.
Maybe. As I say, that's what I say. Not
the last 68 years in Israel, the life expectancy shot up about more than 16 years.
Except the concern, at least in North America, with growing obesity now, is that the life expectancy of the next generation is actually going to decrease.
Yeah, but it's not because of that.
No, not because of this, but that's so.
Painkillers just reduced the life expectancy in the US by 0.1 year over the last 2 years.
Has gone down already.
Yeah, that's because
in the US,
painkillers.
Because painkillers is okay.
The first few years in which the life expectancy went down.
So again, you have the obesity, you have malnutrition, poor food. You have again. I'm not saying that we need to be concerned about this.
What I'm saying is, you take the suntan and drink the water, tap water.
Yeah.
Despite everything, our body manages it correctly.
Let's put it this way. I was asked recently, do I use suntan lotion? And I said yes. I mean, I try to cover up and wear a hat, but the places that are exposed, because the risk from the sunburn is far higher than potential risk of some skin damage from the nanoparticles. I'm willing to take that chance. But like I say, maybe there's nothing to worry about. But on the other hand, as I say, the EPA won't let you drink nanoparticles, but the FDA lets you eat them. So let's have some kind of, sensibility here. We had some other technology we used in nanoparticles to break down atrazine and other chemicals in the ground. It works very effectively. What's the problem? We can't get regulatory approval because it's not consumed. What do you call it? It's a reduction. It's a reduction process. There are enough free electrons from the bacteria. We found a catalyst that breaks down the material very nicely. Nanoparticle catalyst. Okay. What's the problem? It occurs in the anaerobic region. All of the regulatory commissions for agricultural products, you have to do it in there's the soil zone, the first half meter, and then there's groundwater. And that whole region in between simply doesn't exist from the regulatory point of view. So you can't do, you're stuck.
How do you make analysis of nanoparticles?
From the soils, the easiest way in the end, when we had the schmutz solutions, the soil solutions, we simply break them down, do a digestion, usually with nitric acid. Digest it, and then send to ICP-MS.
Yeah, but
so,
how you know what is that part of the nanoparticle and part of dissolved matter?
Oh, because first of all, you have controls, right? So you take the soil just with water, and we do the same acid digestion. Then you do it with the nanoparticles as well. So the increased concentration has to have come from the nanoparticles. Well, you're saying, because you're saying the nanoparticles themselves.
Because also if you introduce nanoparticle to the ICP-MS,
yes,
they give you the same signal like dissolve.
Yeah, but if you want by mass, you can do, as I say, dissolve it. And so that gives you the amount of the ionic form of it. And then from there you can make an estimate.
So you don't make direct measurement of nanoparticles.
We're doing that now, the single particle. Yeah, they generally, most of them, they don't dissolve too much. If you don't do the acid digestion, you get very little dissolution.
I think there is a confusion here. He's talking about experiments where he's adding the nanoparticles.
Yes.
And you are talking about unknown samples which you do not know how.
Ah, okay, now there's an unknown sample that may have, then there's no way of knowing. No way of knowing, you're right. I agree. Except what we can do now, as you see, their initial results, and it depends on the type of nanoparticle, single particle ICP-MS, you can detect. But we found, like, we did it with silver, we were doing work on plant uptake of nanoparticles by plants. And with the SPICP, the single particle ICP-MS, we got silver particles recently, a very clear peak of actual intact silver particles. Copper and zinc, we couldn't find. And we don't know if it's the technique or the plants break them down. So.
I have a question about the last experiment.
What's the last one? Oh, that one, yeah.
Yeah, this one.
Yeah.
How do you know that the silver isn't just being oxidized and then basically it's no longer the same sort of particle? I mean, how do you go about testing the...
Well, from the DLS, when you do the zeta potential and you do DLS, you see that they are aggregates.
Okay.
So, I mean, they retain the nanoparticle form.
Yeah, but they might just be oxidized silver. My question is how do you know that they're just not undergoing some chemical change?
I think, well, we did the electron microscopy on them afterwards too, I think. I know we checked that, I don't remember offhand. Okay. I'll give you an answer on that. Okay, yeah, no, they stay in the nanoparticle form. But there is, they stay as nanoparticle.
They could be silver oxide.
That's possible. If there's an oxide, if there's a change to the surface chemistry, absolutely. But it stays in the nanoparticle structure. Yeah. Okay, then yeah.
So when you did the soil experiment, did you control the porosity and permeability of the material, or?
Well, we measured the porosity. You mean when we took the different soil fractions? Yeah. Yes. Yeah. Okay. So we have the measurements and all of that. But the main point was just, I mean, on one hand you expect as you have the finer grains, the porosity will change, and so you're going to change the retention ability of the nanoparticles. So yeah, we have that information. Okay, thank you.
Those two pulses, those two peaks, actually. Do you see
which peaks?
Those two, the twin peaks that you see during
the breakthrough of particles?
Yeah, with the twin breakthrough peaks. You said that it's because it's clogging and then releasing. Yeah, those. Yeah. Do you see the pressure fluctuations as well?
We didn't.
So you didn't.
Because they are mentality
because you kind of expect. It would be interesting if it correlates or how if there is because if that's the mechanism, then it has to be, or you would expect there to be measurable changes in pressure.
That's the question.
Yeah.
Well, I mean, it depends on the sense.
To kind of, unblock something. If it's mechanical or if it's physical blocking, sure, like you would expect. No, I'm saying it's one way to kind of test.
I agree. The question again is going to be what's the degree of sensitivity, right? To detect the pressure change.
Let's put it this way: if you do see it, it's a positive test in the sense that if you do see it. Sure. Yeah. Yes, that would support. If you don't see it, it's not a.
You haven't
disproved
not that. You just haven't proven that it is. I agree.
Professor Khadash.
Oh, yeah. Oh, you're fake. Mazal tov. Professor, my status.
עוד מעט מן המדע.
אין לי את זה, להקל את זה כבר. מה שלומך אברהם?
אני בסדר. כן, כן, נכון. לא היית בייג'ינג.
לא, חשבתי על העניין של ההוספה של ה-Humic Acid. Humic Acid זה כזה דבר מסובך.
כן.
כימית אתה לא תוכל להבין כלום מה שקורה שם. אנחנו נוסיף חומצות שומן ידועות, או דברים שהם ידועים שאתה יכול לעקוב. סיגל קומפאונד.
כי השאלה, אתה רוצה לזהות משהו ספציפי, או אתה רוצה להגיד, אנחנו יודעים עם שמוץ בחוץ.
שמה?
אנחנו יודעים עם שמוץ בחוץ, נכון? ואז בואו נעבוד עם משהו יותר מציאותי.
לא, ברור, אבל אני אומר, בשביל להבין קצת את המנגנונים הכימיים, אז אולי זה יכול לעזור קצת אם אתה הולך לטיפורות ספציפיות.
No, the time is sort of. So to me, there are only 26